Hello students, this is part 3 of chapter 6 molecular basis of inheritance. Here we will discuss 20 MCQs from previous years PMT based on NCERT core content. Question number 1. If the distance between two consecutive base pairs is 0.34 nanometer and total number of base pairs of a DNA double helix in a typical mammalian cell is 6.6 into 10 to the power 9 base pair, then the length of the DNA is approximately. See the solution. यहाँ हमें length of DNA meters में calculate करनी है. Let us see what is given. Two base pairs के बीच का distance दिया है. It is equal to 0.34 nanometer. Convert this into meters by multiplying it with 10 raised to power minus 9. This is the first step of the calculation. Now, length of DNA calculate कीजिए by multiplying number of base pairs and distance between two base pairs in meters. Means multiply कीजिए 6.6 into 10 to the power 9 को 0.34 into 10 to the power minus 9 से. And the answer will be 2.2 meters. So, option C, 2.2 meters is correct. Mark C as a correct answer. Question number 2. Under which of the following conditions there will be no change in the reading frame of the following messenger RNA? And the sequence of messenger RNA is given in front of you. अगर वन बेस का इंसर्शन हो, जैसे ऑप्शन बी में इंसर्शन ऑफ जी एट 5th पोजीशन, या वन बेस का डिलीशन हो, जैसे कि ऑप्शन सी में मेंशन किया गया है डिलीशन ऑफ जी फ्रॉम 5th पोजीशन, और टू बेसिस का इंसर्शन हो, जैसे डी ऑप्शन में मेंशन किया गया है इंसर्शन ऑफ ए एंड जी एट 4th and 5th पोजीशन, then it would cause change in the reading frame. But if there is deletion of three bases GGU from 7th, 8th and 9th positions given in option A would not cause alteration in the reading frame. Reading frame में कोई change नहीं होगा इस केस में. So option A is the correct answer. Mark A. Question number 3. Which of the following features of genetic code does not allow bacteria to produce human insulin by recombinant DNA technology? Genetic code is nearly universal. What does it mean? इसको एग्जाम्पल से समझिए. UUU कोडॉन फिनाइल एलानिन एमिनो एसिड को ही कोड करेगा सभी ऑर्गेनिजम्स में, वेदर इट इज़ अ वायरस और बैक्टीरिया, ट्री और ह्यूमन बींग्स. जेनेटिक कोड का यही फीचर अलाउ करता है बैक्टीरिया को टू प्रोड्यूस इंसुलिन बाय रिकॉम्बिनेंट डीएनए टेक्नोलॉजी. सो मार्क डी, जेनेटिक कोड इज यूनिवर्सल एज अ करेक्ट आंसर. क्वेश्चन नंबर 4. इफ देयर आर 999 bases in an RNA that code for protein with 333 amino acids and base at position 901 is deleted such that the length of the RNA becomes 998 bases, how many codons will be altered? See the solution. There is deletion of base at position 901. Total bases affected will be 99, that is from 901 to 999. And as you know, a group of 3 bases is equal to 1 codon. So 99 affected bases will be equal to 33 codons. Thus, correct answer is B. 33 codons will be altered. Mark B as a correct answer. Question number 5. Which of the following is not a property of the genetic code? A property देखिए, non-overlapping. Yes, this is one of the property of genetic code in which each nucleotide is a part of one codon. Means एक न्यूक्लियोटाइड 2 एडजेसेंट कोडॉन्स का पार्ट नहीं हो सकता, एक न्यूक्लियोटाइड एक ही कोडॉन का पार्ट होता है। बी प्रॉपर्टी देखिए, एम्बिगुअस, दिस इज नॉट अ प्रॉपर्टी ऑफ जेनेटिक कोड। जेनेटिक कोड इज नॉन एम्बिगुअस, मीन्स वन कोडॉन कोड्स फॉर ओनली वन एमिनो एसिड, एक कोडॉन एक ही एमिनो एसिड को कोड करता है। सी प्रॉपर्टी देखिए, डीजेनरेसी, यस code is degenerate, means some amino acids are coded by more than one codon. Now see the D property, universal, the code is nearly universal. So out of 4 given properties, option B, ambiguous, is not the property of genetic code. So mark B as a correct answer. Question number 6, whose experiments cracked the DNA and discovered unequivocally that genetic code is triplet. Genetic code is deciphered by Nierenberg and Mithail. So correct answer is option D, mark D. Question number 7, which of the following pairs of a codon is correctly matched with their function or the signal for the particular amino acid? Out of 64 codons, 3 codons UAA, UAG and UGA are stop codons. They do not code for any amino acid. Thus, out of 4 options, option D, UAG and UGA are stop codons. This is the right match. So, mark it as a correct answer. Question number 8, what would happen if in a gene encoding a polypeptide of 50 amino acids, 25th codon UAU is mutated to UAA? If 25th codon UAU is changed to UAA, then translation will stop after the formation of a chain of 24 amino acids because UAA is a stop codon. So correct answer is A, a polypeptide of 24 amino acid will be formed if 25th codon UAU is mutated to UAA. Question number 9, after a mutation at a genetic locus, the character of an organism changes due to change in mutation, sequence of nucleotides changes, which further causes change in the sequence of amino acids in a protein and result in change in protein structure. So correct answer is option A, protein structure changes due to mutation which results in changes in the character of an organism. So mark A as a correct answer. Question number 10, if the DNA codons are ATG, ATG, ATG and a cytosine base is inserted at the beginning, then which of the following will result? As insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion, here cytosine is added in the beginning. See the lower picture showing the change after addition of cytosine in the beginning. It will be now CAT, GAT, GAT, G. There is a change in the reading frame and this change is mentioned in option A. So mark A as a correct answer. Question 11. The first phase of translation is, see the solution. First phase of translation is activation of amino acid in the presence of ATP and linkage with its particular transfer RNA. This process is called amino acylation of transfer RNA or charging of transfer RNA. This process is completed in the presence of enzyme called amino acyl transfer RNA synthase. So correct answer is C. The first phase of translation is amino acylation of transfer RNA. Mark C as a correct answer. Question number 12. A complex of ribosomes attached to a single strand. A complex of ribosomes attached to a single strand of messenger RNA during translation is called as polysome. So, correct answer is C, mark C. Question 13. The two subunits of ribosome remains united at a critical ion level of. See the diagram. Each ribosome is made of two subunits, a smaller subunit and a larger subunit. These two subunits are associated with the help of magnesium ions. So, answer is option A. Magnesium ions are required to unite two subunits of ribosomes. Mark A as a correct answer. Question number 14. Match the following genes of lac operon with their respective products. See the diagram and match. A. I gene, it is responsible for formation of a product repressor protein. B. Z gene, it is responsible for formation of beta-galactosidase. C. A gene, it is responsible for formation of transacetylase. And D. Y gene, and it is responsible for formation of permease. So, correct answer is D, mark D. A matches with 3, B matches with 1, C matches with 4 and D matches with 2. Question number 15. Select the correct match. Option A, B and C are wrong matches. See the option A. Alex Jeffries in 1984 invented DNA fingerprinting. B. Alfred Hershey and Martha Chase proved that DNA is genetic material using bacteriophage. C. Matthew Michelson and F. Stoll proved semi-conservative mode of replication in bacterium E. coli. Now come to option D. It is a correct match. Francois Jacob and Jacques Monod proposed lac operon concept. So mark D as a correct answer. Question number 16. All of the following are the part of operon except. See the diagram. Option A, operator. Option B, structural genes which are lag Z, lag Y and lag A, and option D, promoter. These are the part of operon. But option C, an enhancer, it is not the part of operon. So answer is C, mark C. Question number 17. Which of the following is required as inducer for the expression of lac operon? See the diagram. In lac operon, lactose is the inducer. It binds with repressor protein formed by I gene of operon and inactivates it by forming repressor lactose complex. It allows RNA polymerase to bind with promoter and transcription proceeds. So, correct answer is A, mark A. Question 18. Lac operon is an example of inducible operon because when lactose is added, it induces lac operon to express its genes. So, correct option is D, mark D. Question number 19. Select the two correct statements out of 4, 1 to 4 statements given below about lac operon. See the statement 1. Glucose or galactose may bind with the repressor and inactivate it. This is wrong. It is lactose which binds with repressor protein and inactivate it. Statement 2 पर आइए. In the absence of lactose, the repressor binds with the operator gene. Correct statement है ये. अगर लैक्टोज एब्सेंट है तो रिप्रेसर ऑपरेटर जीन से बाइंड कर जाता है एंड ऑपरॉन ऑफ हो जाता है. स्टेटमेंट 3 पर आइए. द जेड जीन कोड्स फॉर परमिएज. नो, दिस इज द रॉन्ग स्टेटमेंट. जेड जीन कोड्स फॉर बीटा गैलेक्टोसाइडेज. कम टू स्टेटमेंट 4. दिस वॉज एल्यूसिडेटेड बाय फ्रैंकोइस जैकब एंड जैक्स मोनार्ड. This is the correct statement. So, Correct option is C. Statement 2 and 4 are correct about lac operon. So, mark C as a correct answer. Question 20. Which enzyme will be produced in a cell if there is a nonsense mutation in LAC-Y gene? See the diagram. अगर LAC-Y gene में mutation हो गई है, then only one protein B-galactosidase from LAC-Z will be produced. Rest permease from LAC-Y and Transacetylase from LAC-A gene will not be able to produce. So, correct answer is C, mark C. So, that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.